Right. So Dr. Jia's title of his talk is Disease Mechanisms Underlying Neurodegeneration Caused by RNA Metabolism Abnormalities.
Thank you. So at first, I would like to thank either the McGovern Institute at MIT side and the organizer to invite us here and communicate each other and present my current work here. So I just recruited by Tsinghua University about a year. So today my talk is majorly focused on some work we are going to work on. So, recently there are multiple evidence suggest that RNA dysfunction of RNA processing may play a key role in many neurodegenerative diseases. For example, the SMN1 molecule that involved in biogenesis of the pre-MI splicing building block has caused the spinal muscular atrophy. And the other evidence from the dysfunction of RNA binding proteins, such as the IgH-mu binding protein, we know that that the molecule may have a function on RNA or DNA helicase. And the other famous one is TDP43, and originally identified by Virginia Lee and John Trojanowski group. And they involve in the mutation has been found in familiar and sporadic ALS and FTD patient. And the other evidence from the repeat DNA repeats insert into the long coding region in some gene that the function is not that clear, and that people believe is a gain of function. So the question I think remain in the field is why the dysfunction of RNA processing cause neurodegeneration. So my postdoc work somehow also contribute to the evidence. So we use forward genetics approach to study neurodegeneration mechanism. So in other words, it's a phenotype-driven approach. So we started with some mutant mice with neurodegeneration. And for example, you see here the 7-month animal in the cerebellum region, the neuron is gone compared to the wild type. And after we find, or we characterize, we identify the mutant animal, we further identify the mutant gene, or the modifier that can enhance or suppress the pathogenic pathway. So this kind of work will deepen our understanding of disease mechanism, because if fly can model disease or learning memory, like Dr. Yichang said, so our mouse, because mouse is mammal, and there have like more than 90, their gene have more than 90% similarity to human genes. So this finding will help us to deepen our understanding of disease mechanism. So one particular case today I'm gonna share with you is one mutant mice. This is generated from EMS-induced mutagenesis. And the mice have a particular neurodegeneration in the cerebellum. So majorly is the granule neuron. So in 4 months, the homozygous mutant, most of the cerebellum granule neuron is gone compared to the wild type litamate. And we also found the neuron degeneration in the other brain region, including hippocampal cortex and the spinal cord. So the nature of neurodegeneration is clear caspase-3 dependent apoptosis. So that I believe you can appreciate that by the caspase-3 staining and the tunnel staining. So the next question we want to ask is what the mutation, what the gene dysfunction cause that phenotype. So using the classical mouse mapping, we narrow the mutant interval to 2 MIT marker, and it's contain 10 protein coding gene. And we sequence every one, and we didn't find any polymorphism between the wild type and mutant. The other feature in our interval is there are 5 non-coding RNA, like cluster, RNA cluster in our interval. So one of them, I need to point out, just one of them have a 5 nucleotide deletion. So what is this non-coding RNA? So this non-coding RNA we call is U2 small nuclear RNA. That is the basal component of pre-mRNA splicing. So the RNA splicing can be separated to 2 steps. One is the assemble phase. So at that moment, so the other uridine-rich snRNPs. So here is the exon 1 and exon 2. In between is the interference sequence. So the exon is the protein coding sequence. So during the splicing, the interference sequence we also called intron will be spliced. So at the assembly phase, the U2 and the U1 responsible for recognize the 5' splicing site. And the U2 will base pairing recognize the branch point site in the intron. And the adenosine is the key nucleotide. The hydroxyl group will attack the 5' splicing site. So the U2 hold the branch site close to the 5' splicing site and fulfill the first catalytic reaction. So where is our mutation? So actually the 5 nucleotide is very close to the U2 and the branch side duplex. The U2 sequence is recognized the branch side in the intron of a transcript. So the 5 nucleotide, 2 of them is in the branch side recognition sequence, and the other 3 is in between 2 domains. One is branch side U2 duplex, and one is the U2 U6 helix 1A. The U6 is in green, and is another uridine-rich snRNA involving pre-mRNA splicing. So one question puzzles us a lot is why U2 is a basal component that causes specific neurodegeneration. So when we check the expression profile of this mutant, and we found, so here I want to point out this assay is RNase protection assay. No matter how we did that, but here is the Y-type form. So that means, so the U2 have multiple copies because it's a housekeeping gene and have the essential function. So in our interval, at least have 5 copies. So in the mutant, homozygous mutant, there are Y-type. But here is the mutant form. So the mutant form actually is very high in the cerebellum. That is the majorly affected brain region compared to the cortex and the spinal cord and the other non-neuronal regions. And we also, so today, because the time limitation, we also showed that this mutant U2 actually is temporally regulated. That means after postnatal 30 days, the mutant U2 is upregulated in the cerebellum. So the next question we would like to ask is how this mutant U2 affect splicing. So actually at the beginning, we checked a lot of splicing events, but we didn't find any major defect. Then we think maybe some highly regulated splicing events, like alternative splicing, may be affected. So we go back to find 20 neuronal alternative splicing events, and we check it. And at this time, 10 out of 20 is majorly affected. So here I just show you one case. So this 3' axon intron is from the one a neuronal gene called L1 camp. So the intron 27 actually is a small intron. It actually is an alternative spliced intron. So why we see that? Because in the Y type, the intron is retained no matter what time point, P20, P30, and P40. The intron 27 is retained. But intron 28 is constitutive spliced. We never see intron 28 signal. But in this mutant, after P30, as I mentioned before, this mutant is upregulated after postnatal 30 days. And this intron retention is dramatically, it's become the major form compared to the spliced form. And and we also check the intron 28, the constitutive splicing events. We didn't see any difference between Y type and the mutant. So this data suggests that this particular U2 mutation affect some type of alternative splicing, but not constitutive splicing. So after that, we want to globally check the splicing defect in this particular mutant, in the cerebellum. So we find using the RNA-seq data, next generation sequence, high throughput sequence, we found many alternative splicing event change. But here I want to emphasize, or I want to draw your attention is the intron retention events. So every dots showed here is the intron retention events. The y-axis is the intron retention intensity. So it's the log 10 value. So that means 2 is 100 folders increase. So these events in the mutant cerebellum, there are 4,000 retained intron events in the mutant. But in the Y type, there are only 300 events that are significantly retained in the Y type cerebellum. And so the X show, so we also distribute these events by their intron length. And very surprisingly, we found that the intron retention events, almost half of them are small introns. So that means this event is susceptible to this particular particular U2 mutation. So we believe that our data provided the definitive evidence suggests that the dysfunction of RNA processing may play a key role in the etiology of neurodegeneration, together with the other findings. So this work is also very important for us because it provides an ideal model to study how the RNA metabolism causes neurodegeneration. So back to the disease pathogenic pathway. So in general, the disease mutant will through some pathogenic pathways such as here I simplified ABC gene pathway cascades then cause the disease. But in our human population, so someone is sensitive to some disease, but someone are not. So there are some like genetic variants or modifiers that can influence or enhance or suppress the pathway, then will affect the disease onset and development. So that is also true in the inbred strain. So the original NM291 mutation actually is a B6 background. But when we cross it to the other strain, and we found this strain, actually is a BOB6 strain, can largely attenuate the disease manifestation. So suggest there is a dominant modifier that can attenuate the disease. So to identify this modifier will help us to understand how and why the RNA metabolism abnormality results in neurodegeneration. And we believe. So the other strategy we want to do is, if some mouse model can model the disease, so whether we can use ENU-based mutagenesis, increase the genetic variance in the mouse model, then we can define or identify the modifier that can accelerate or suppress the disease development. That will help us to understand the disease mechanism. But when we back to check the mouse model, that model the neurodegeneration caused RNA metabolism, we are very frustrated. So many models, they are transgenic. They are random insert into the genome and have multiple copies in the genome. For example, TDP43. So right now, there are transgenic models available. But we know that Y-type, even you overexpress Y-type TDP43, also have the neurodegenerative phenotype. So one of the RNA binding protein draw a lot of our attention is IGH-mu binding protein 2, because this mutation is a recessive mutation. Actually, it's a loss of function mutation. And it's causes type 1 spinal muscular atrophy with muscle weakness and motor dysfunction. And patients have this mutation often died after birth because of the respiratory failure. So why we interested in this molecule? So here is the structure of this molecule. So it has the RNA DNA helicase domain and one like single RNA recognition sequence and a protein interaction sequence domain. And here showed by now there are more than 60 mutations, human mutations, has been identified. So here showed some of the mutation in these genes. So luckily we have a mouse mutation. Also we have mouse mutation. So the mouse mutation here showed at this position there is a splicing variance. So this splicing variance cause the splicing jump into the intron region, then cause the molecule. In the animal, only 20% of the full-length molecule is left. So this particular mouse mutation have very similar phenotype as human patient. So this mouse mutant also a recessive mutation, and it's have the motor neuron degeneration and died very early after birth. So this mouse model could be a very nice model for us to screen the genetics modifier that can attenuate or rescue the phenotype. Our strategy is use these mutant mice and use ENU-induced mutagenesis to screen any dominant modifier that can rescue or attenuate the disease manifestation. So hopefully, our screening will help us to identify a lower gene that can help us understanding disease mechanism caused by loss of function of IGH-mu binding protein 2. So I would like to use these slides to summarize our research pipeline. So we use forward genetics to study neurodegeneration, majorly that caused by RNA metabolism abnormalities. And we also want to construct some reliable models that can model the neurodegeneration caused by RNA processing abnormalities by using recently developed TALEN or CRISPR technologies. This work is working in my current lab. So I would like to end my talk with my acknowledgement slides. So Dr. Susan Ackman is my postdoc mentor. And John Wu from Stanford helped us to analyze next generation sequence. And I would like to thank constructive discussion from our colleagues, both from Beijing McGovern Institute and Tsinghua McGovern Institute, and our animal facility, and Dr. Jian Zhongxi and Bao Zhang help on the TALEN and CRISPR technology, and my lab member in the Tsinghua University. Thank you very much.
Yeah, we're doing very well on the time, so we have a few questions. Okay, we have time for questions.
Thank you very much. Wonderful talk. One quick question is that you use the RNA, the metabolic, approach to study. Is it only restrict to the cerebellar degeneration? Does it also apply to the cortex?
Oh yeah.
So you focus on the SMA, on the cerebellum, instead of the, the say frontal lobe, some other gyro in the cortex.
So actually, as I mentioned, so this particular U2 mutation is highly expressed in the cerebellum. But we also see the phenotype in the hippocampal and the cortex and the spinal cord, but much less. That means the dosage play a very important role. If we can dose this mutation in the spinal cord, we expect we will see motor neuron degeneration.
One last quick thing is that, have you tried to expose the system to a immune, dysregulation, for instance, like the chronic inflammation model, against the background and the context, and to see how the RNA metabolism would change accordingly?
Yes, it's something we are thinking, but we haven't done yet. Yeah.
So I like your suggestion that this could be, that many of these neurodegenerative diseases could be a defect of a splice variant or splicing.
Yeah.
And so that suggests that a lot of the ones that have been around for a long time, like SOD, and that people are looking at, that they should actually look at the regions that are affected first and then do RNA-seq to see what's happening.
Yeah,
right?
the
Because
question is the cell type specificity. So
well, frequently you can figure out what
sure.
types of cells are
Sure.
beginning to degenerate first. So you figure you go back a couple of weeks or something.
Yeah,
but,
sure.
But that also requires that RNA-seq become much less expensive, because if you have to look at different ages or different cell types,
sure.
and it's a real issue. I don't know if China's looking into making it more accessible for people without giant budgets and things like that.
Yes, so far the RNA-seq right now everywhere, I think globally, is become more and more less expensive. And the other is so no input RNA-seq. So like less than 10 nanogram.
Oh, really? So
Yeah,
even
yeah,
that's
yeah,
normal.
something like that will dramatically improve our
that's right.
sick
'Cause
consequence.
what you're suggesting is that you not only have to look at a specific area, but you have to look at a specific age.
Yeah, specific cell type, and yeah, so yeah, sure.
Is it possible, and this is sort of a leap from what you were talking about, but I was stimulated by the title of your talk. Is it possible that the RNA metabolism for complex, you're talking about quite a neurodegeneration and quite a heterogeneous group of illnesses with quite a different heterogeneous cascade of effects. Is it possible that some combination of the medication and the cascade of events that accompanies a neurodegenerative disorder changes the RNA metabolism?
So I didn't get your question. So what? Sorry. Yeah.
I'm not a geneticist. I'm a,
Sure.
a psychologist. I guess. So, my question is: so your study was looking at whether or not the metabolism of the RNA affects the neurodegenerative process. I've worked with a lot of people with neurodegenerative disorders, and I guess I find myself wondering whether the I don't know very much about RNA and metabolism, but I'm wondering if the metabolism is affected by the cascade of different events, including the treatment, rather than just the abnormalities in the RNA, the genes affecting the RNA. Because most of the neurodegenerative diseases I know, and I was part of a team that looked at schizophrenia for a while, no one thinks about those kinds of illnesses as due to single genetic inputs, but in fact a multiple number of inputs and also environmental factors. So I guess that's why I'm thinking about the causes behind the
Yeah, it's
RNA
complicated. Yeah, so one thing I think we need to think about is there could be multiple targets. So like here, there are thousands of targets affected. And intron often introduce premature termination codon and introduce some like truncated form, truncated protein. So that may also contribute to the disease development. So it's complicated. So at this moment, we are not very sure the disease. So for the particular mutant U2, so disease is caused by RNA toxicity or protein toxicity. We are not very sure, yeah, at this moment. But I prefer it's RNA toxicity. Yeah.
Guess we have to move on.
Thank you.